Okay, today we're going to go over plant structure and organization. So we've already looked at the different cell types and the meristems that create them, as well as some of our tissue organization. So mostly what we're going to be looking at today are the roots and the shoots, and looking in detail at each one. You will need to get out your MONOCOT versus DICOT stems and roots worksheet, because you'll be filling out some of that. And we have a lot to cover today, so I'm going to try to get through it pretty rapidly. Some of the slides I will not be narrating. It'll just be for you to review some of those and look at them on your own. But yeah, we're going to move rapidly today. Shoots and roots. All right, let's get started. Okay, so our root system obviously is the first part that develops in the embryonic stage of a plant. It's the first part that emerges from the seed, and its purposes are to anchor the plant in the soil and to give it support. And we've talked quite a bit about how it helps with water absorption and minerals. And also, one of the things we really haven't mentioned is that it also functions as food storage in certain plants, such as in sweet potatoes. Not white potatoes, just so you know. So you know the difference between those. But in sweet potatoes are roots. So every root has 3 zones. There is the zone of cell division, zone of elongation, and the zone of differentiation. So on our image here, when we talk about the root apical meristem, that's going to be right here. That is where the zone of cell division happens. This root cap is mostly for protection. It does serve another purpose that we're going to get into in detail much later. But for our purposes, we're looking at these 3 zones right here. And so as the cells divide, and you can think of the maturation process moving in this direction. Okay, so we have new cells developing here down at the meristem, which is in the zone of cell division. And then as they start to mature, they become longer and longer. And then even further back, as they mature, they begin to differentiate into certain types. And the zone of cell differentiation is where you're going to find the root hair cells. Okay, so our root tissues in a Udicot, we're going to start there. And some of this will look familiar, so I'm going to go ahead and just give you the terminology here, and then we'll look at what it looks like on the next slide. So we have the epidermis. We've talked about that quite a bit. It's that single protective layer on the outside. It's also where you start to have specialization into root hairs. The cortex, which kind of makes up the bulk of the root, and that's just loosely packed parenchyma cells. Water moves around and through these cells towards the center of the root. That's also where, if there's going to be food storage, that takes place in the cortex. And then there's the endodermis, and that's a single layer of cells, and that's between the cortex and what we call the vascular cylinder. And it plays a vital role because it is responsible for the regulation of water and ions into the vascular tissue, so into the xylem, so it can be moved up and down. And it has what's called the Casparian strip, and we're going to talk in detail about that endodermis, so you need to get to know that one pretty well. And then going further into the center of the root is what we call the stele, and that involves the pericycle, which is that first layer of cells within the vascular cylinder, so just inside from the endodermis, and that's where the lateral roots come out. And then, of course, the rest of the stele is made up of all our vascular tissue. So this is what it looks like. So, like I said, we start with the endodermis. That's the layer, single layer on the outside. That's the protective layer, and it has differentiated cells such as root hairs. The cortex is this whole area right here. Those are the parenchyma cells that can do food storage, and water comes through this area when it's being absorbed from the root hair through the root hairs. And then just inside of that cortex is the endodermis. And the endodermis has a Casparian strip. What that means is that each cell within the Casparian strip has a basically a waxy coating. And that waxy coating forces water to go through the cell itself of the endodermis, and that's where the water regulation happens, is right through here. So water can't go around because of the Casparian strip. Okay, coming in from that, the next layer in is the pericycle, and that's where lateral roots develop. And then we get into the stele. That's this whole section here. And that means that we have vascular tissue in there. Oh, sorry, the stele also includes the pericycle. Sorry, pericycle and vascular tissue is the stele. Okay, so I mentioned the Casparian strip, and that's within the endodermis. So there are two basically different pathways that water can enter the plant through the root. We know that our root hairs are the places for absorption. So two things can happen. Water can go into the cell, into that epidermis cell, and then travel through plasmodesmata going from cell to cell to cell. It can also, let me go ahead and say that's the symplastic pathway, sym kind of being within. Apoplastic or apoplastic means that the water comes on the outside of the cell wall of the root hair, and then basically between all the cortex cells, but then it hits our Casparian strip. Remember we said that water couldn't go past that. So if you look up here, this is what that Casparian strip looks like. It's on the outside edges, on the transverse walls, and it's just a waxy coating, so that if you are going through the apoplastic pathway, you get stopped at the endodermis, and then the water is forced to go in through the cell, and that's where regulation happens, and that is the apoplastic pathway. So that Casparian strip is made of lignin, and it's also made of suberin. Suberin should sound familiar because suberin, remember, is also part of the cork, and that is a block to water because it's impermeable to water. So just reviewing those two different pathways, because you will need to know them for sure. Our red is the symplastic, and that's what goes inside each cell and then gets to the endodermis, again goes into the cell, and the endodermis is what decides how much water and how many ions are being let through, and then it continues into the stele and down into the xylem, where it gets transported throughout the plant. The blue is the apoplastic pathway, and that goes between all the cell walls until it hits the endodermis, where it is forced to go into the cells, and again that regulation happens there. Okay, so we've talked about the epidermis and the cortex, and we've gotten to the endodermis, where that water regulation takes place, going further inside to the root. Now next we come to the pericycle. It is the single layer, first layer of what we call the stele, so it is right next to the endodermis, and this is where lateral roots are formed. So if you look at these images here, here's our epidermis, this is our cortex, we have an endodermis, and the next layer in is our pericycle, and that is where the lateral root is emerging from. This is just a cross section, and it comes further out and goes that way. Okay, so the very center of our root, and remember we are focusing on eudicots right now. So we've gone through the epidermis, the cortex, We've gone through the endodermis and then the pericycle where we had lateral roots arise from. So just to give you a little orientation to where you are in this cross-section picture, this would be our cortex. And we have the endodermis here labeled in red. The next layer in would be the pericycle, and that's where our lateral roots arise from. And so the pericycle, along with the xylem and phloem, make up the stele. So in a eudicot, in a root of a eudicot, there's this star pattern of vascular tissue. And that's how you know when you do a cross-section, you know that it's a eudicot in the root because of the cross-section looks like a star. So all this red here is the xylem tissue at the very center. And then this section here, this is the phloem that goes pretty much in between the xylem branches. Okay, so that's what it looks like in a eudicot root. So we looked at the three different zones. We had our meristematic or cell division zone, we had the elongation zone, and the maturation zone. Now I just want to take a minute to look at the root cap because it actually plays a very significant role. The very first thing that it does is it protects that meristematic region because roots have tip growth, so it's continually pushing through the soil, and if it didn't have a protective coating, it would just knock off that meristematic region and not be able to grow anymore. So that's the first role that it plays. It also plays a role in sensing gravity, and that is the purpose of these columella cells, which is these right here. And they're called that because they really are more columnar, kind of elongated. So in this image here, this would be our meristematic region. So looking really closely at that meristematic region, our apical root meristem that we've talked about, we have the quiescent center, which is just a few cells, which would be probably about right here. And that portion is a portion of the apical meristem that doesn't divide very often. That usually only happens if there's been a big injury to the root. And then just below that is what we call the initials, or it would be similar to stem cells in animals, and that's where all the division happens. Below that is going to be our root columella cells. So in this picture, all these right here are root columella cells. Okay, so here we're looking a little bit closer at our columella cells, and the reason I wanted to make sure that you know the different structures here is because in a little bit, in another lecture, we're going to talk about gravitropism, and this is going to be important information for you. So in this image, we have our columella cells. This is basically several rows of them. I think this one has 3 to almost 4. The 4 right here will probably slough off pretty soon, so they may not play much of a role. But within the columella cells are special organelles called amyloplasts, or they're also called statoliths. And they're right here. And they don't have any color to them, but they are responsible for starch synthesis and storage. And so what they do is they store these very large starch grains, and you can see them in each of these cells. And if you notice, they are all at the bottom of the cell. So I'm circling the amyloplast. This right here is the entire columella cell, and note that the amyloplast is at the bottom. And the reason for that is because they are very heavy, so they fall in whichever direction is down. So we'll get to details on that later, but I did want you to know what those are. So far we've been talking about dicot roots, and Remember we said that if we did a cross section of a dicot root, we could tell that it was dicot or eudicot, sorry, because of this star or cross pattern. And remember that's going to be the xylem, and then we would have phloem in between like that. Okay, that's what we've been looking at this whole time. If we looked at a monocot root, the center would look very different. In this case, we have circles of xylem, and then phloem is in between the xylem like that. And we have something in the monocot root called a pith that we don't see in the eudicot roots. And that pith is just made up of parenchyma cells, and it can be for food storage. Everything else about the root is the same. So we still have the epidermis, we still have the cortex, we still have the pericycle for water regulation. Pericycle for lateral root growth. We still have the endodermis that regulates water entry into the stele. But the stele looks very different between a eudicot root and a monocot root. So make sure you can tell the difference between those two. All right, so let's just walk through the layers together that we've been looking at. And this time we are looking in a monocot root. So to start with, we have our epidermis at the very outside for protective coating. And remember those, if we were higher up in the root, we might see some root hairs if it was in the zone of maturation. And then we have the cortex, which is where most of the food storage takes place. And then next is our endodermis, and that's where the Casparian strip takes place. Just inside of that is the pericycle, where our lateral roots come. And so then we have xylem. In this case, we have some large xylem right here. There might be some smaller ones, but see how there's like a circle, a cylinder of circles all around right here. Those are all xylem. And then we have the phloem right here. And then inside is this pith that we don't see in our dicots. So this is the worksheet that you all have, and now having gone through each of those layers all the way from the outside in, I want you to take out just a pencil and lightly fill this out. See if you can do it on your own first, and then the next slide has all the answers. So see if you are right or not. This is a good practice. So now that you know what monocot and dicot roots look like at the cellular level when we do a cross section, looking at the whole plant, how would you tell the difference? Okay, on the left we have monocots. Monocots typically have a fibrous root system. So that means that each of the roots comes from the junction with the stem, so straight down like this. And that is even true here in some of our palms, if you can see that. In our dicots or eudicots, they have typically a taproot with lateral roots that come out from that, or just our straight taproot. Now, there are all kinds of modifications of roots. So just real quickly, I mentioned that sweet potatoes are root tubers. So remember, that would be where those parenchyma cells are storing a lot of starch. There's also aerial roots. So if you've ever seen cypress knees or mangrove trees, they have these roots that come up out of the water. Those are called aerial roots. And then we also have support roots. If you've ever seen a banyan tree, usually this is in a more tropical climate, but there are roots that come straight from the stems down here just to support, because these trees get very large, so there's extra support with these types of roots. All right, let's move on to stems. So our stems have our terminal bud, which contains the shoot apical meristem, and then has these bud scales that protect the bud. And then here's our stem. And each year when growth resumes in the spring, these bud scales fall off, they leave a scar, and then growth continues in that direction. So you can tell how old a plant is by looking at the terminal bud scars, so the bud scale scars. So here's one here, and then another one would be forming here once the growth starts. Or if you look at the side branches, we know it started here, and we are on to the first year right there. So our stem, some of this you have seen before. We have our shoot apical meristem right here, and that's where all of the primary growth is going to take place. Of course, we have our lateral apical meristems as well. And so something that may look new, these right here are our leaf primordia, or our immature leaves. They will mature into full leaves. Our ground meristem right here, that is what produces the pith and the cortex. So something new is this center section of the dicot stem has a pith. So this is not to be confused with our monocot root has a pith. Remember, the dicot root does not, but the dicot stem does have a pith, and that's where food is stored. The cortex area, which is going to be right here, that's what's green in the stem. That's where photosynthesis takes place. Now, our procambium gives rise to primary vascular tissue. Okay, so this is going to be our bundle of xylem and phloem. And here, as the stem matures, then the other meristem that we've talked about before, called the vascular cambium, develops between the mature primary xylem and the primary phloem. So if we look at an enlarged view of this, here is our primary xylem and our primary phloem, and then what develops in between is the vascular cambium. And remember, that vascular cambium is going to produce secondary xylem to the inside and secondary phloem to the outside. Okay, so here is a cross section of our eudicot stem, and you can see the pith in the middle that we talked about, and the ring of vascular bundles around the outside of the stem. So you definitely have to know if you are looking at a stem or a root in cross section, because if you have a ring like this of vascular bundles in the roots, it's going to be a monocot, but in the stems it's a eudicot. And between the xylem and the phloem layers is the vascular cambium. So that's starting right here. And so if we look over here, we have the xylem and we have the phloem right there, and then this layer is the vascular cambium. The vascular cambium is unique to dicots. So if we think about our monocots, if you remember, those are mostly, if you think about them as grasses, you know that grasses don't produce, they're not woody. Remember, secondary xylem is actually what we call wood. So it's real stiff, and you don't see that in grasses. So a vascular cambium is only present in eudicots, not monocots. So here is a cross-section comparison of the monocot stem versus the eudicot stem. And note the absence of a vascular cambium in the monocots. And the vascular bundles in monocots are spread all throughout the stem. So you do have some on the outer edge, but you also have them all throughout the center. So there's no distinction between the cortex and the pith in the monocot. If you look at the dicot stem cross-section, we have this large pith in the middle, and note that there's no vascular bundles in it. And then the cortex would be out here. And there's this lovely ring of vascular bundles. And as the plant matures, of course, we have the vascular cambium that develops between the primary xylem and the primary phloem. So I'm going to scoot over to this photo on the right, and this is a monocot stem, because you note that there are vascular bundles all throughout. There's no distinct pith. And then we're just going to take a look at one of these vascular bundles. Typically speaking, the portion that's facing the inside, the center of the root, contains the xylem. So we have large air spaces, and then we have large vessel elements, and then the portion more towards the outside, or to the outside of the root, didn't say that, towards the outside of the stem is the phloem. So if we look at just one singular bundle, this section towards the inside is the xylem, and the section towards the outside is the phloem. Okay, and that's in our monocot roots. Our xylem is always towards the inside, whether it's monocot or dicot, and the phloem is always towards the outside. But the way those vascular bundles are arranged throughout the tissue of the stems is different between monocots and dicots. Okay, so stems themselves can be either herbaceous or woody. So what is the difference? I've said this a couple times, but just driving it home a little bit, only eudicots can be woody. So when we're talking about monocots versus dicots, only angiosperms are either monocots or dicots. So that's where that division takes place. So of angiosperms, only eudicots can be woody. Now, our conifers can also be woody, and conifers, remember, are gymnosperms. So those are the two types of plants that can be woody, either angiosperms that are eudicots or gymnosperms that are conifers. Those are the types of plants that can be woody. And woody refers to a specific type of growth. So herbaceous plants have only primary growth, right? So those are derived from the primary meristems. Woody plants have primary growth, and they additionally have secondary growth. So when we talked about those secondary meristems or lateral meristems, that's only found in woody plants. So our vascular cambium and our cork cambium are only found in woody plants. So looking at our woody stem, and this is at maturity of a woody dicot stem, and it's going to look very different from an herbaceous dicot stem. But there are three distinct areas within a woody stem. So first we have what we call the bark, and the bark has living phloem, and what is also referred to as the periderm, which we talked about before, which is the cork cambium and the cork that it produces. So bark has cork cambium, and living phloem. Second, we have wood, and this shown here is the heartwood and sapwood, which is secondary xylem. So that's produced by this vascular cambium right here. The vascular cambium produces secondary xylem this way, which is called wood, and secondary phloem this way, which is part of the bark. And then the third part of a woody stem is this pith in the center. Remember, we said the eudicot stem has pith in the center. All right, so let's talk about growth rings, because you're very familiar with wood. When we cut down a pine tree, this image to the right is pretty much what it looks like. So I can't say enough that wood is secondary xylem. Okay, that's all it is, secondary xylem. So how is a growth ring produced? Okay, so in trees with a growing season, the vascular cambium is dormant in the winter. Okay, because it's too cold, there's not much water availability, it's dormant. When water is present in the spring and the tree starts to be active again, new secondary xylem is laid down by the vascular cambium. And when there's lots of water present, there are large vessel elements in that xylem. So in our image here, let me make sure I get my little thing going. We have springwood. You see how large those vessel elements are? That's produced in spring because of lots and lots and lots of water. As summer approaches and water is more scarce, the elements are narrower and have thicker walls. So that's right here, our summer wood. And then fall concludes the season with very thick walled fibers. So all right here. And then in the winter it's dormant again. And then spring starts again, and here we go with the second part. But that, with the spring wood and summer wood and then the dormant season, that creates this pattern that gives you an annual ring. Okay, so if you can think about it, if our vascular cambium is right here, as it produces xylem that way in the spring, it'll lay all these down first, and then summer it'll lay down these, and then fall, and then winter, and it stays dormant. So it produces that ring. So that's an annual ring. So we know how old a woody tree is based on how many annual rings it has. So this primary xylem right here doesn't really count, right? So here's 1, 2, 3. So this particular tree would be 3 years old. So looking at your worksheet having to do with a woody stem, I want you to try to figure out which each of these layers should be labeled as, and then the next slide will give you the answers, but you will have to understand how this is. And if you need to ask me in lab, we can go through this again. I'm happy to do that. So do you remember that when we were talking about xylem, we said that it is dead at maturity? Our secondary xylem is kind of broken down into two different parts within a woody tree, especially as it ages. The first is what's called the sapwood. And that's this lighter section here. And that's the xylem that is still active in transport. As a tree gets older, and that same sapwood, as we start putting more and more layers towards the outside here, this sapwood would look like that on the inside. And that darker part here is it's what we call the heartwood. And in older trees, that's wood that's no longer functioning in transport. And in order to prevent infestations or infections, the tree plugs those vessel elements with resin to make sure that bugs don't make a home. So if you've ever heard of fat wood, which lots of people use for starting fires, that is the heartwood of a pine tree because it has all that resin that can be highly flammable. Okay, so this is just to give you an idea of a little bit of stem diversity, some modifications to stems. So tendrils on grapes, for instance, are a modification of a stem, and that allows grapevines to grab onto structure. The second is a rhizome tuber. So a rhizome, if you remember, is an underground stem. So white potatoes are actually tubers that form in those rhizomes. And so that's just the opposite of a sweet potato that is a root storage tuber. White potatoes are actually stem tubers. And then just another example is a stolon, is what we call it, in a strawberry plant. If you've ever seen them throw out these stems, and then from those stems roots start to grow and they start a whole new plant. So those are just a little bit of some stem diversity. Okay, the last thing we're going to talk about is leaves. We've talked about the root system, and we've talked about the stems, which are part of the shoot system, and the other part of our shoot system is the leaves. So let's get into that. This is where the majority of photosynthesis takes place. Okay, our plants tend to be divided based on some qualities of their leaves. Particularly to begin with, we group them into evergreens or deciduous plants. Evergreens are those that bear their leaves the entire year, and they tend to have needle-type leaves. So our pine trees, our Fraser fir, they have needles all year round. And then our deciduous trees are those that lose their leaves at the end of the growing season. For example, this sugar maple. So I purposely picked Fraser fir as our evergreen to look at because it's very important to North Carolina. North Carolina Christmas tree industry is ranked as the second in the nation in the number of trees harvested. Everybody loves a good Fraser fir. And of our Christmas tree industry, 96% of all the Christmas tree species are Fraser fir. And if you look at this map, they are only grown in a limited amount of counties in the western part of the state, mostly in the northwestern part. Allegheny, Ashe, Avery, Buncombe, all those on my list over here. So for evergreen trees in general, they either have to live in warmer climates or have a type of like antifreeze type of substance in their leaves, and that helps them helps the cytoplasm to not freeze in the winter. In addition to that, they have very waxy coverings to their leaves, and also tend to be needle-shaped leaves, and that's because it limits the amount of margins that the leaves have, and therefore not a lot of exposure to the cold environment. So why do deciduous trees lose their leaves? Usually it's thought that it helps to conserve water and energy. Most deciduous trees have broad leaves that have more margins, so they're susceptible to damage and cold and dry weather. So if they shed their leaves, then they allow for the tree to conserve that water and energy. An additional benefit is in the spring, most deciduous trees will flower before they leaf out, and so those leaves aren't hindering the pollinators from getting to the different flowers. So how do deciduous trees lose their leaves? If you can get this down, your friends are going to be impressed because people always ask that. How do they lose their leaves? How does that work? So here you go. Once you get this, you're going to impress all your friends. So there's a process. And it starts with what we, well, first of all, let's say this. When trees lose their leaves in the fall, we call that abscission. And the start of that process is the tree reabsorbing all the valuable nutrients from the leaves. And in particular, it starts with reabsorbing the photosynthetic pigments and those organelles because those contain the most energy. So usually it will start by taking out the chlorophyll, and once they do that, the green comes out of the leaves, so you start to see the orange colors, and that's just because the carotenoids are left, or the anthocyanins. Those are different pigments that your leaves already have, but they're overshadowed by all the chlorophyll. So as those different pigments are starting to be reabsorbed, you start to get the beautiful colors that you're used to seeing. So when the anthocyanins are produced, that's what acts as that lower freezing point, sort of the antifreeze for those cooler months, those fall months. And then, interestingly enough, what we call abscission cells form a bumpy line where the petiole of a leaf attaches to the branch. And what that basically does is it says, okay, I'm cutting you off. I'm done with you. I've gotten as much nutrient out of you as I need. So at the end of the abscission process, that abscission layer basically grows over and kicks off the leaf. So that is how deciduous trees lose their leaves. So we've been talking a lot about the differences between monocots and dicots, so we want to do that with leaves as well. So the leaf morphology is very different between dicots and monocots, and the placement of the vascular tissue within the two is very different as well. So let's start just by looking at the leaves themselves. So on this slide, the leaves on the left, the oak and maple leaves, these are dicots. And the one on the right, those are monocots. So the first thing to look at is where the veins lie, the vascular tissue. So in our dicot, we have this netting type effect. So we've got a main vein as well as branching veins that come off of that, and even veins that come off of our branches. So that's a netted effect. In our monocots, we have parallel veins. So they just go like this, all along the blade. The other thing is that dicots have what we call a petiole. You may have noticed that I mentioned that when we were talking about leaf abscission. Here's the petiole. That is the section that attaches the leaf blade to the stem itself. That's called a petiole. Monocots do not have a petiole. The blade itself just wraps around the stem and acts sort of like a sheath. At the cellular level, our dicot and monocot leaves look very different as well. So let's look at the structure of a dicot leaf to start with. To begin with, we're going to talk about what we call the mesophyll cells. The mesophyll cells in a dicot leaf are of two different types. On the top side of the leaf, just under the epidermis, and in this one you can see the cuticle as well, is what we call the palisade mesophyll cells. Palisade is easiest way to think of that is that there are these long stretched cells like you would see in a palace, okay? And all of the mesophyll cells is where we're going to have the chloroplast, so that's where the photosynthesis takes place. So we have palisade mesophyll cells on the top, and then on the bottom side of the leaf, or towards the bottom side of the leaf, is what we call spongy mesophyll cells. So spongy mesophyll cells have a lot of air spaces in between them. Now, why would that be? Okay, remember when we talked about how our dicot leaves had guard cells and stomata on the underside of the leaf and not so much on the top? Well, if we are expecting gas exchange through these stomata only on the underside of the leaf, we have to get that air or that carbon dioxide through to the whole leaf. So even though we can do photosynthesis in these spongy mesophyll cells, and that's great because they're close to the stomata, so they already get the carbon dioxide, we have to leave air spaces so that that carbon dioxide can then be transported up to the palisade mesophyll cells, where we can also have photosynthesis. Okay, so that's what that looks like. And remember that we have these veins of plant tissue in our plant tissue, in our leaves. So here is a vascular bundle. There's another one here. It may also be running in that direction because this is our dicot leaf. In our monocot leaf, this is a cross section. So if you'll remember, when we had our parallel veins, if we took a cross section of it like this is what it would look like. So here is a vascular bundle, here's another vascular bundle, here's another vascular bundle. And if you can reach back in your memory and remember that we have stomata and guard cells on the top and bottom of our monocot leaf. So for instance here, that's actually a pair of guard cells, as is this right here. And so our mesophyll cells are not divided into palisade and spongy mesophyll cells. It's mostly just spongy mesophyll cells all throughout. Okay, so that's the difference between a dicot leaf and a monocot leaf. So some variations on our leaves in cactus, they can be modified to be spines. Sweet pea tendrils, if you've ever had sweet pea plants, those tendrils are actually not stem modifications like they are in the grapes, but they are leaf modifications. And then of course we have our lovely Venus flytrap, where we have hinged leaves so that it can be a carnivorous plant. So this is the summary of how to tell the difference between a monocot and a dicot plant. So do take your time to pay attention to these and note the difference. I will not hold you liable for the pollen slits and pores. You don't really need to know that, but the rest of it is pretty important. So I'll be able to show you a plant and you'll be able to tell me if it's a monocot or a dicot. All right, I'll see you next time.